Hello students, welcome to the bio point. Yes, now we shall start a new topic that is known as Human Genome Project. Or in short, we can say HGP, a project which is related on human DNA basis. So, first we know what is genome. First we should know what is genome. A genome, a complete set of genes present in an individual. A complete set of genes which is found in an individual, it called genome. Understand? We know that it is a nucleus. In the nucleus, chromosomes are found, and in that the DNA is found, and on that DNA we have already told you that base pairs are found, that A, T, G, C, A, T, G, C. Such type of base pair found means in the DNA base pairs, that is A, T, G, C are found. Understand? And we know that in human about 3 into 10 to the power 9 base pairs are found. Understand? So to take knowledge, to know the knowledge about that, and to study about that, a huge project is formed. So such a huge number of base pair, and in your book you will see that if the cost of one pairs will be $3 for its maintenance, the total will be about $9 billion. So you see, and if a page contain 1000 watt base pair, then we can write about so many books, we can write so many pages. So it's a huge project, and it was first started. This project was first started in 1990, 1990 means actually this is 1st October 1919, the first day, and it is continued up to, it was 2003. So the time period was 13 years. The total time period was, and the project was started first in United States. United States and later many other countries like China, Japan and Australia, like that, they starts to join with that project. Now what is the goal of HGP? That is goal of Human Genome Project. Means goal means uddesh. What is the goal of? Now the goal of HGP to determine the base pairs that makes the human DNA. What base pairs which are helpful, which makes the DNA, to know about that, and also to identify and mapping all the genes which are present in human genome, and how to identify them and to mapping them, to take knowledge, to know knowledge about that is its goal. Second, that it is started by concrete venture, and the main thing, the association or the company or the concern which was started this project, that is Department of Energy and National Institute of Health of US. They help this project, and in the 1990 identify the approximate about 20,000 to 25,000 genes in human DNA. They identified such for their project. Determine the sequence of 3 total base pairs which is found in human DNA. In the humans DNA of humans, we know that about so much base pair is found. What their sequence, what their arrangement, to know about that. Second, the information which we get, understand, we should store in database and improve the tools for data, and the machine or technique should be improved to analyze this data, and this was ethical, legal and social that may arise from the project. These are some issues, that ethical, legal and social issue which may arise in the project, and that study, that thing give a new branch of study, that it leads a new branch of study that is called bioinformatics. That is called bioinformatics. Now next one is methodology. We have studied at the time of transcriptions that DNA changes into, DNA undergoes 2 approaches. Means in such a way that is codon, non-code, codon, non-code, code, non-code. That is exon, intron, exon, and intron. 2 types of the codons, that is in the heteronuclear, that is in the heteronuclear stages, that is in the, and that codon parts undergo, that forms the messenger ribonucleic acid, and then they take part in the formation of protein synthesis. Understand? So DNA appreciation 2. One is codons, non-codons. They take parts, by means of translations, and they take part in the protein synthesis, but they do not go. There is no importance. Now the first step is to sequence the only codon part. Means in that the only codon part will be sequenced, and that is called expressing sequence tag, means EST. And the second, to sequence the both codon and non-codon part, that is called sequence annotation. So these are the two methods of sequences. One is only the codon parts, and now next one is the both the codon and non-codon part. Now the first DNA first we extract the DNA, means that's extraction. Second, this extract DNA cut into fragments. Suppose this is the DNA, this cut into fragments. Now they colon in suitable host with a specific vectors. Now they coloned with, means undergo coloning, and that coloning takes place in the suitable host. Understand? This suitable host may be bacteria or yeast. Understand? And the specific vector will be BAC or YAC. BAC, that is bacterial artificial chromosome. This is yeast artificial chromosome. And after the cloning, after cloning means with the host cell, the sequence DNA we get. So in this way we say that after the extraction and fragmentations, and these are colonized with in suitable host, that host may be bacteria or yeast, and for that the specific vector we use, this vector may be BSC or YSC. BSC, bacterial artificial chromosomes, and that is yeast artificial chromosomes. They undergo host undergo host, and they sequence the DNA, and that DNA sequenced in the host cell, and thus finally we get the sequenced DNA. At salient features, human genomes contain about so much base pair, you know. Generally, the genes consist of 3000 bases and its variable number may be varies. The function of about 50% of gene we ate on. Function of about 50% genes are ate unknown, and the chromosomes 1 has the maximum gene so much and the Y has the lowest gene. Now the application, and there are also so much silent features in your book NCRT. The application to diagnose the genetical disease and treat, means by diagnosing the genetical disease we treat by this project. Second, treat the disease by using DNA technology. By using DNA technology, we can treat it by this human. So these are the some salient features and the application of the Human Genome Project.